So now we are discussing about the sizes and shapes of bacteria. Bacterial display a wide diversity of shapes and sizes. Bacterial cells are about one tenth the size of eukaryotic cells and are typically zero point 0.5 to 5.0 micrometer in length. However, a few species are visible to the unaided eye. For example, Thiomargarita namibians is up to the half a millimeter long. Euplopsicum facialsoni reaches zero point 0.7 millimeter and Thiomargarita magnifica can reach even 2 centimeter in length, which is 50 times larger than other known bacteria. Among the smallest bacteria are members of the genus Mycoplasma, which measure only zero point 0.3 micrometer, as small as the largest viruses. Some bacteria may be even smaller, but these ultra micro bacteria are not well studied. If we discuss the shape of bacteria, most bacterial species are either spherical called cocci or rod shaped called bacilli, and some bacteria called vibrio are shaped like slightly curved rods or comma shaped. Other can be spiral shaped called spirelia or tightly coiled called spirochetes. A small number of other unusual shapes have been described, such as star shaped bacteria. This wide variety of shapes is determined by the bacterial cell wall and cytoskeleton and is important because it can influence the ability of bacteria to acquire the nutrients attached to the surfaces, swim through the liquids and escape the predators. If we will discuss about the multicellularity, most bacterial species exist as single cells. Others associate in characteristic patterns. Neisseria forms diploid, streptococci form the chains, and staphylococci grouped together in bunch of the grape cluster. Bacteria can also group to form larger multicellular structures such as the elongated filament of Actinomycetota species, the aggregate of Myxobacteria species, and the complex hyphae of Streptomyces species. These multicellular structures are often only seen in certain conditions. For example, when starved of amino acids, Myxobacteria detect the surrounding cells in a process known as quorum sensing, migrate towards each other, and aggregate to form fruiting bodies up to 500 micrometer long and containing approximately 100,000 bacterial cells. In these fruiting bodies, the bacteria perform separate tasks. For example, about 1 in 10 cells migrate to the top of the fruiting body and differentiate into a specialized dormant state called a myxospore, which is more resistant to dying and drying and other adverse environmental conditions. Biofilms. Bacteria often attach to surfaces and form dense aggregation called biofilms and larger formation known as microbial mats. These biofilms and mats can range from a few micrometer in thickness to up to a half a meter in depth and may contain multiple species of bacteria, protists, and archaea. Bacteria living in biofilms display a complex arrangement of cells and extracellular components forming secondary structures such as microcolonies, through which there are networks of channels to enable better diffusion of nutrients. In natural environments such as soil or the surfaces of plants, the majority of bacteria are bound to surfaces in biofilms. Biofilms are also important in medicine, as these structures are often present during chronic bacterial infection or infection of implanted medical devices. And bacteria protected within biofilms are much harder to kill than individual isolated bacteria. If we discuss the cellular structure of bacteria, intracellular structure, the bacterial cell is surrounded by a cell membrane which is made primarily of phospholipids. This membrane encloses the contents of the cell and act as barrier to hold nutrients, protein, and other essential components of the cytoplasm within the cell. Unlike eukaryotic cell, bacteria usually lack large membrane-bound structure in their cytoplasm, such as nucleus, mitochondria, chloroplast, and other organelles present in eukaryotic cells. However, some bacteria have protein-bounded organelles in the cytoplasm which compartmentalize aspects of bacterial metabolism, such as carboxysome. Additionally, bacteria have a multi-component cytoskeleton to control the localization of proteins and nucleic acids within the cell and to manage the process of cell division. Many important biochemical reactions such as energy generation occur due to concentration gradients across membranes, creating a potential difference analog to a battery.